called an honorary enzyme. Okay, we talked about negative feedback inhibition. Again, often through allosteric inhibition of the product to an allosteric site on the enzyme that's at the beginning part of the pathway. Okay? That's the most economical way to conserve resources, such that when you have enough sufficient product, don't make a lot of intermediates you don't need. Turn it off at the beginning here. That makes sense, right? Economic sense for the cell. Okay? So the product acts as an allosteric inhibitor. Okay. All right. So those are the 5 ways that cells can control enzyme activity. Now let's look how some physical parameters can affect the activity of an enzyme, specifically temperature, pH. Okay. About the influence of pH, and we'll start with pH on enzyme activity. Okay. Now, numerous amino acids that make up proteins, there are numerous amino acids that are ionizable. Okay, part of the R group can become charged or not, and that often will depend upon the pH of the environment. Okay. For example, if you had an R group, let me show you an ionized amino acid here. Partly ionized. Okay. If an R group, and I see it may be exactly correct, but I think this is an R group here. It's got an additional carboxyl group here. The pH is low. That means there are a lot of protons in solution, right? It's an acidic environment, and they're going to be attracted to negative charge here and bind, eliminating that charge. Okay, and if those charges are important enough to maintain tertiary structure, as they often are, this can cause the protein to begin to change its conformation, depending on the pH environment, right? Or if the pH is elevated, if the liquid's a little too basic, well then the protons are less abundant, and this becomes charged again. Okay, so ionizable amino acids respond to the pH environment, and that can, because a large part of the tertiary structure depend upon these interactions amongst R groups, especially among ionizable R groups, pH can affect the conformation. And if you affect the conformation, that can either stabilize the active site, so it accepts substrate, or it can warp out of shape. Right? So pH is critical to maintaining protein structure and function because of its influence on these ionizable amino acids that make up proteins. Does that make sense? Okay. I mean, I can pull up the amino acids, we can look at those, but you can do that yourself. Okay. And so here's a classic example. We look at 2 enzymes. There's a number of enzymes that help us to digest proteins into the individual amino acids. Protein digestion begins in the stomach. There's an enzyme released from certain cells forming the inner lining of the stomach. One of them is called there's an inactive form produced called pepsinogen that's activated by the acidity of the stomach. The acidity of your stomach is around pH 2. Okay, under that acidic environment, the pepsinogen, an inactive precursor of this protein digesting enzyme, is cleaved into pepsin, and that pH stabilizes the active site, so it can begin to break down polypeptides into shorter protein chains. Okay? Now, when your stomach begins to pass, when you get this acidy, partly digested mass of food in your stomach, it's called chyme. I'll spell that for you in case you've never heard of that. The chyme then moves from the stomach into the small intestine through a pyloric valve at about a volume of about a tablespoon, gradually. It's not all shoved into the small intestine because the small intestine lacks the mucosal barrier that protects the stomach lining from this acidity down in the stomach. In the small intestine, the pH is 7 or even a little more basic than that, 7 to 8. Okay? So if this massive chyme were to move into the small intestine, it would burn the small intestine, cause some serious damage there. So it's sent bit by bit, okay? And in the small intestine is where protein digestion is completed, where proteins are completely broken down to individual amino acids so they can be absorbed through those co-transport pathways we've talked about, right? Talked about co-transport, where sodium-potassium pump builds a sodium gradient outside that diffuses back in to a co-transport, can drag a sugar or amino acid in with it. Remember that? Okay? Right? So to complete protein digestion in the small intestine, it needs a different set of enzymes. One of those is called trypsin. There are a number of others. Okay? And these enzymes work best at a basic or near neutral pH, which is the environment in which they occur in. Okay? And so depending on the environment where the enzyme is destined to work, the ionizable amino acids will be under the appropriate status to allow this enzyme to work at a pH of 2, but at a more basic pH. So when this chyme moves into the small intestine, this enzyme becomes inactivated as the environment of the small intestine approaches a pH of near neutral or even slightly basic. And now enzymes that could never work in the stomach now are activated. Okay? So all enzymes are sensitive to pH and their effects on the conformational shape via the actions of pHs on the ionizable amino acids. Okay? That make sense? And your body puts a lot of effort into regulating the pH of your fluids through buffers. There's the blood buffer system, there's the cellular buffer systems to help to regulate the pH environment so the enzymes work appropriately. Okay? Does anybody know any fluid compartment in the body where there is no buffering system and those tissues are very sensitive to pH changes? Any organ that you might imagine is very sensitive to pH changes? So the cerebral spinal fluid has no buffering capacity. So it protects the brain, the fluid, the cerebral spinal fluid that surrounds the brain, it protects that from changes in the acidity. The other fluids is a blood-brain barrier that keeps, tightly regulate the cerebral spinal fluid because the brain fluid has, the cerebral spinal fluid, or the brain fluid, has no buffering capacity. But cellular fluids, cytosol has buffers. Blood, of course, has the bicarbonate buffer system. Okay. All right. And then, of course, temperature is another physical parameter that can affect protein enzyme structure. It can lead to denaturation. So again, animals are susceptible to extreme temperatures, in part because extreme temperatures cause protein denaturation. And if enzymes denature, they don't work, and then processes break down. Okay. Now, again, remember that a chemical reaction increases with increasing temperature. You all know that. And for most enzyme catalyzed reactions in the body, from 0 to 40 degrees Celsius, within this range here, the rate of reaction doubles with every 10 degree C increase. Okay? It's called the Q10 effect. We're going to double the metabolic rate, increase the temperature 10 degrees Celsius. And so with increasing temperature, you've got increasing reaction rate. Even if it's enzyme catalyzed, it's going to work faster and faster and faster and faster. Right? Between 0 and 40. Now you get much beyond 40, now things start to go bad. What happens now beyond 40? Why is the reaction rate now dropping off? Look how steep the curve is here. A nice steep curve here. Again, every 10 degree increase, I double the reaction rate. Double. You see that? When I get beyond 40, 45, and now the rate of decline is even steeper. Because of what's happening here. What's happening to the protein, the enzyme? At high temperature, it means it's really shaking violently now, right? It's going to physically shake itself out of shape. It's going to denature. Okay? And so again, animals have to strictly regulate body temperature, okay, to maintain enzymes in this best zone here. Okay? That's what makes high temperature so dangerous. Okay? And we warm-blooded mammals, okay, we're called homeotherms. We're endothermic homeotherms. Those are two words you should know for the pre-lab quiz on Thursday. Endothermic homeotherms. Those, they're not really mean the same thing. Anybody know a synonym for homeotherm? What's the common, if I say that animal's a homeotherm, what would the common usage someone would say? Well, that animal is warm-blooded. Okay, what does that mean if you're warm-blooded? Say it again. Well, body temperature, but you maintain a constant body temperature except in extreme conditions. So we mammals and birds are homeotherms. We keep a pretty steady state body temperature. For we mammals about 37 Celsius, birds are a little higher. And regardless of the environment, of course, it breaks down at extremes. You can go into hypo or hyperthermia, but across a broad range of environmental temperatures, your body temperature is constant. And it's elevated. Okay, it's elevated at a high temperature, much higher than a cold-blooded poikilotherm. Okay, now that's different from the word endotherm. So endotherm, a lot of people kind of mix the two together, endotherm and homeotherm. Homeotherm, your body temperature is constant.